us today to discuss Context Therapeutics. My name is Sue Romanoff, Head of Healthcare Content at Edison Group. We're very excited to be working with the Context team during this pivotal time for the company. On today's webinar, we have an impressive lineup of key opinion leaders to discuss some recently announced data from Context Therapeutics. Following this presentation, we'll host a Q&A session. If you'd like to submit a question at any point, please do so by using the Q&A box at the bottom of your Zoom application. We can flip to slide 2. Before we begin, I'd like to briefly pause on this forward-looking statement slide. I'd also like to note that today's webinar is being recorded. We can go to slide 3, please. As you can see here, we have a full lineup today. Presenting, we have Martin Lear, Chief Executive Officer of Context Therapeutics, Dr. Evan Dick, Senior Vice President and R&D at Context Therapeutics, Lauren Werner, MD and PhD candidate, Elisabetta Margoni, PhD, Antonio DeRosso, MD and PhD, and Joseph Rucker, PhD. With that being said, I'll hand it over to you, Marty.
Thanks, Sue. Pleasure to connect with everyone today, and thank you for joining the webinar. If I can ask you to move to the next slide. Joining me on this call today is my colleague, Dr. Evan Dick. For those of you who are not familiar with Evan, he's had a wonderful long career that spans academia, large pharma, and for the last 2 decades or so, small and emerging pharmaceutical companies where he's led R&D at most of those organizations. Today we'll be speaking about our 2 lead programs, onapristone extended release, which we also refer to as ONA-XR, our oral progesterone receptor antagonist that is the subject of multiple ongoing clinical trials, 3 phase 2 trials and 1 phase B/2 trials that are described here on this slide 5. As well as our emerging preclinical bispecific antibody program targeting Claudin-6 and CD3. It's been an exciting time for our company. We went public in Q4 of 21, and this year of 2022 is really shaping up to be a pivotal data year for our company, where we'll start turning over some of the data cards of our various clinical trials, presenting updates on those trials starting this summer through the end of this year. Next slide, please. So from here, I'm turning it over to my colleague, Dr. Evan Dick, our head of R&D.
Thank you, Marty. Hello, everyone. I'm happy to have you with us. This slide summarizes our 2 programs, our clinical stage program, which is called ONA-XR for onapristone extended release. As you saw from the prior slide, it's currently in phase 2 development in multiple studies. The basis of the reason for going into this is that progesterone receptor oncogenic signaling is associated with breast and gynecologic cancer. Onapristone is a progesterone receptor antagonist that suppresses progesterone receptor oncogenic signaling. The extended release is a proprietary oral form and has potential for combination with selective estrogen receptor degraders, CDK4/6s, and a number of other inhibitors and drugs that are relevant in the treatment of breast and gynecologic cancers. We have exclusive worldwide ownership with the exception of ex-Greater China. We also have a preclinical stage program, as Marty mentioned, which is a bispecific antibody development program targeting Claudin-6, which we'll speak about later when Joe Rucker speaks, and the T-cell engaging arm is CD3. Claudin-6 is an oncofetal protein. I don't want to steal Joe's thunder, so I'll let him speak more about that. We have high selectivity for Claudin-6 versus its closest paralog, Claudin-9, and it looks like we have excellent selectivity relative to our competition, and we have a worldwide license to Claudin-6 from Integral Molecular. The next slide, please. We have 4 terrific candidates speaking today, and I'll introduce them as I introduce their presentations. May I have the next slide? Our first speaker is MD PhD candidate Lauren Warner from Christy Hagan's lab at the University of Kansas Medical School. Lauren's current work falls within the field of activating anti-tumor immune responses in immunologically cold carcinomas, which mammary tumors are an example of cold carcinomas. Lauren's work probes why and how progesterone receptors might be targeted to promote immune-mediated anti-tumor activity in breast cancer. Lauren, I give it to you.
Thank you very much for that introduction. So today I'm very excited to present to everyone some key data highlights from the poster that we presented at the AACR Annual Meeting this past week. Next slide, please. So I wanted to just start by introducing why we care about the immune system in cancer. So the overall goal of the project that I'm working on in the Hagan lab is to determine if progesterone modulates the immune system to promote breast cancer growth. And the overall reason why we care about the immune system in cancer is because our immune systems are the key defense in preventing tumor growth. Shown on this slide is a type of an immune cell called a T cell attacking a cancer cell. And if the immune system is functioning properly, our immune systems are able to eliminate cancer cells before they grow into tumors. So in order for cancer to grow in the body, there has to be some disruption of this system that allows cancer cells to hide from our immune systems. Next slide, please. Our immune systems are also very important when it comes to our responses to various types of cancer therapy. And one of the key types of cancer therapy where our immune response matters is a class of therapy called immunotherapies. These are therapies that work by activating our immune systems to better detect and attack cancer cells in the body. And what we know about patients when they're treated with immunotherapies is that patients typically fall into kind of one of two classes. There are patients that respond poorly to immunotherapies, that's shown on the left-hand side of this slide, and patients that respond well. And what we know about the different types of patient responses to immunotherapies is that it really depends on the makeup of their tumor. So shown on the left-hand side of this slide is something that we have defined as an immune cold tumor. These tumors are known to have a poor response to immunotherapy. And what causes that is that there's actually low numbers of immune cells that are present within these tumors. So something is keeping the immune cells out of these tumors. They're not there to detect and attack and eliminate tumor cells. The types of immune cells that are present in these tumors are usually suppressive types of immune cells, such as regulatory T cells, which is the purple cell shown on that tumor. And then on the right-hand side of this slide is depicted an immune hot tumor. These are tumors that have high numbers of infiltrating immune cells, so lots of immune cells present in the tumor to detect and eliminate tumor cells. And these tumors are known to have a good response to cancer immunotherapies. And unfortunately, when it comes to hormone receptor positive breast cancer, so breast cancers that express estrogen receptor and progesterone receptor, largely these tumors fall into the category on the left-hand side of this slide, which is the immune cold tumor subtype. Next slide, please. So the goal of my project in the lab has been to try to determine if progesterone, which signals through the progesterone receptor, is contributing to hormone receptor positive breast cancers having an immune cold tumor microenvironment. And if that is true, we hypothesized that we could use anti-progestin therapies such as onapristone in order to convert these immune cold tumors, which don't have lots of immune cells present, into immune hot tumors, which are shown on the right-hand side of this slide. Having more immune cells present means that these tumors could have an improved baseline anti-tumor immune response and could be better responders to immunotherapies. Next slide, please. So the first experiment that we did with onapristone to try to determine if it could be used as an agent for breast cancer treatment was to do a dose response. So shown on this slide is a tumor growth curve where we treated mice that had PR expressing mammary gland tumors with either 3 doses of placebo or 3 doses of onapristone. Onapristone is shown in the red and pink, and the placebo is shown in blue. And what you can see on this slide is that 2 different doses of onapristone, the 12 milligram and 30 milligram doses shown in the red and light pink, were able to almost completely inhibit the growth of these PR expressing mammary gland tumors. Next slide, please. So another experiment that we did early on was to compare head-to-head onapristone with another antiprogestin to see if different antiprogestins would have differential effects. So shown on this slide is a tumor growth curve where we grew PR expressing mammary gland tumors treated with either placebo, shown in blue, or onapristone, shown in pink, or mifepristone, shown in red. And what we found in this experiment was that both antiprogestins were able to almost completely inhibit tumor growth. Next slide, please. Another thing that we did in these experiments where we were measuring tumor growth was at the end of the experiment, we collected the tumors and measured the numbers of different types of immune cells that were present within these tumors. And what we saw was that one specific type of immune cell that we're very interested in, called regulatory T cells, which are a suppressive type of immune cell that prevents anti-tumor immune responses. This cell type was actually decreased in the tumors that were treated with onapristone. So this provided us with the first piece of evidence that perhaps onapristone can actually help convert immune cold tumors into immune hot tumors by decreasing the numbers of suppressive types of immune cells present within these tumors. Next slide, please. So an additional experiment that we did was to try and determine whether the ability of onapristone to inhibit tumor growth is truly mediated by the immune system of these mice. So shown on this slide is our original experiment on the left, and an experiment that we did in immunocompromised mice shown on the right. And the purpose of doing this experiment was to see if these mice don't have an intact immune system, are we still able to achieve control of tumor growth with onapristone? And what you can see when comparing these 2 graphs is that in our original experiment, where the mice had intact immune systems, we achieved almost complete tumor growth inhibition with onapristone and mifepristone. But if you look at the graph on the right, which was done in the immunodeficient mice, which lack a functional immune system, you'll see that onapristone, which is shown in pink, only was able to inhibit tumor growth to about 50% of the level that we saw in the mice with an intact immune system, suggesting to us that, at least in part, the ability of onapristone to inhibit tumor growth requires an intact immune system. Next slide, please. An additional experiment that we did to try and determine which cell of the immune system is most important for contributing to the effect that we see with onapristone was the experiment shown on this slide. And in this experiment, we depleted one specific type of immune cell in the mice. These cells are called CD8 T cells, and they are one of the main types of immune cells that are involved in anti-tumor immune responses. So one of the main cells involved in killing tumor cells. And in this experiment, we treated mice with either placebo or onapristone with the addition of either a control IgG antibody or an anti-CD8 T cell depleting antibody. And what we saw first was that both of the groups treated with onapristone again had significantly decreased tumor sizes. But the exciting piece of data that I wanted to point out was actually a comparison between the 2 onapristone treated groups. Next slide, please. So if we zoom in on the groups that were treated with onapristone, we have our control antibody treated group shown in purple and our anti-CD8 depleting antibody treatment group shown in blue. And what you can see is that the mice that were treated with these depleting antibodies had significantly increased tumor growth compared to the mice that got the control antibodies, suggesting that if you deplete CD8 T cells, which are one of the key types of cells involved in anti-tumor immune responses, we don't get as good of tumor growth inhibition with onapristone, suggesting that this cell type is very important in the response to onapristone. Next slide, please. So to summarize my talk, I wanted to just go over the key points that I presented to you today. Next point. So first, previous data from our lab has suggested that progesterone promotes tumor growth by suppressing the immune system in PR-positive breast cancers. Next point. So we first showed today that onapristone was able to completely inhibit the growth of PR-expressing mammary gland tumors and reverse the effect that progesterone had on immune cell infiltration, converting immune cold tumors into more immune hot tumors. Next point. We also showed that complete growth inhibition with onapristone required the mice to have an intact immune system. Next point. We also showed that growth inhibition by onapristone was diminished when we depleted CD8 T cells. And finally, overall, we think that our studies provide a very strong rationale for continuing to investigate onapristone as an agent to both increase baseline anti-tumor immune responses and to potentially enhance the response of tumors to immunotherapies. So with that, I'd like to just recognize the former and current members of our lab. Next slide, please. As well as our funding sources. So thank you very much for your attention.
Thank you, Lauren. That was great. Very good. On the next slide, our next presenter is Dr. Elisabetta Marangoni of Institut Curie. Dr. Marangoni works extensively with patient-derived xenograft models, or PDX models. The development of PDX models has raised the level for how well animal models may predict drug responses in the clinic. Dr. Marangoni will tell us about her PDX studies combining onapristone with established antiestrogens, as well as with a CDK4/6 cell cycle inhibitor that works downstream of estrogen receptor drive. Elisabetta, over to you.
Hello, thank you very much for this invitation. So today I will present a summary of a project that we perform in our institute, Institut Curie. And the title of this presentation is Targeting Progesterone Receptor with onapristone in Patient-Derived Xenograft Model of ER-positive and PR-positive bone metastasis of breast cancer. Next slide. So progesterone receptor is expressed in approximately 70% of breast cancer. It can be activated in an hormone-dependent and also independent ways. And finally, it also interacts with the estrogen receptor and is a target of estrogen receptor-dependent transcription. Next slide, please. So the objective of this work were first to determine the anti-tumor activity of onapristone combined with endocrine treatment, CDK4/6 inhibitors, and P3 kinase inhibitors in patient-derived xenograft model of ER-positive and PR-positive breast cancer. And secondly, to analyze the transcriptional modification in onapristone-treated tumors. Next slide. So first, we evaluated the expression of progesterone receptor by Western blot analysis in different PDX models that were available in our group and that were either established from the primary breast tumor or from bone metastasis of patients that were treated in the adjuvant setting by endocrine treatment. And by this kind of analysis, we could identify different models that show a significant expression of progesterone receptor, especially in the bone metastasis derived models. So we choose to use the model 1117 and the model 1101, and you can see that 1117 tumors has a very high expression of PR, while the 1101 has a lower expression of PR. Next slide. So the clinical history of the 2 patients that were associated to these models is summarized in this slide. You can see that both patients were treated by chemotherapy in the early stage setting, in the neoadjuvant or adjuvant setting. And then they receive different type of endocrine treatments such as tamoxifen or aromatase inhibitors in the adjuvant setting and also in the metastatic setting. At the genomic level, the 1101 model has a typical profile of ER-positive breast cancer with amplification of the cyclin D1 and also focal amplification of FGFR1 receptor. While the 1117 model has a mutation, a pathogenic mutation in the P3 kinase gene and amplification or copy number gains in PAC1, cyclin D1, and cyclin B1. And importantly, an immunohistochemistry analysis was performed on the bone metastasis biopsies that was engrafted in the mice to generate a PDX model. And the expression of progesterone receptor was quantified by a pathologist, and the results are concordant with the Western blot analysis of the PDX models, with a low score of 4 for the 1101 model and a PR score of 8 for the high PR expression model. Next slide. And here you can see the picture of the ER and PR staining in the 2 bone metastasis biopsies, 1101 and 1117. And next slide, please. So what we did is to treat this PDX model in vivo with onapristone given alone as compared to fulvestrant, which is an endocrine treatment, and as compared to different combination, fulvestrant and palbociclib, which is now the standard of care for metastatic breast cancer, and different combination including onapristone, such as fulvestrant combined to onapristone, and the triple combination fulvestrant, onapristone, and palbociclib. In this model, which is the low PR expressing model, we found no effect of onapristone alone. We found a tumor growth inhibition of onapristone combined to the CDK4/6 inhibitor palbociclib, and we found no benefit of adding onapristone to the endocrine treatment fulvestrant given alone or associated to palbociclib. Next slide. Conversely, in the PR, in the high PR expressing model, the 1117, we found a weak effect of onapristone given alone. Again, we found a tumor growth inhibition of onapristone plus palbociclib. But importantly, we observed an increased response of the triple combination of fulvestrant, onapristone, and palbociclib when compared to fulvestrant alone or to the double combination. And it's important to note that in this triple combination group, almost all xenografts were in tumor regression under the treatment. Next slide. And in this xenograft model, which is a P3 kinase mutated tumor, we also tested the combination of onapristone with the P3 kinase inhibitor alpelisib. And we observed that onapristone increased the response to fulvestrant and alpelisib, and that onapristone combined to alpelisib was as efficient as fulvestrant combined to alpelisib. Next slide. Next, we wanted to investigate what kind of transcriptional modification can be observed in the tumors treated by the different treatment group, onapristone alone as compared to fulvestrant and the double or triple combination. And to do this, we perform an RNA-seq analysis of the model that was the most PR expressing model, 1117. And here you can see the gene set enrichment analysis of the xenograft treated by onapristone alone as compared to xenograft in the control group. And you can see that we found different gene sets that were enriched in this kind of analysis and that were associated to proliferation or meet targets for the downregulated genes. And next, we perform the gene set enrichment analysis of the double combination fulvestrant palbociclib as compared to control, and we found an upregulation of interferon response and downregulation of different cell cycle progression gene set. Next slide. Then we analyzed the group treated by the triple combination fulvestrant, onapristone, and palbociclib, and the gene set enrichment analysis identified again upregulation of genes of the interferon alpha or gamma response, tumor necrosis factor or KRAS signaling, and downregulation of different gene sets associated to cell cycle progression. Next slide. And when we compare the triple combination to the double combination at the gene expression level, we found an increased upregulation of different interferon and inflammation pathways, a downregulation of estrogen response, and upregulation also of apoptosis and p53 pathways. Next slide. And here you can see as example the heat map of the late estrogen response and apoptosis hallmarks, where you can clearly see a downregulation of different genes in the triple combination group in the late estrogen response heat map, or upregulation of many genes associated to apoptosis on the right of the slides. Next slide. And finally, we wanted to investigate whether onapristone could affect ER-PR interaction by performing a dual link proximity ligation assay, and by this with this analysis we could see that the treatment by onapristone alone does not affect ER-PR interaction. However, a significant decrease in ER-PR interactions was observed in tumors treated by the triple combination as compared to tumors treated by the double combination. Next slide. So in summary, onapristone increased the response to fulvestrant and palbociclib in 1 PDX of metastatic breast cancer with high expression of PR. Treatment by the triple combination lead to downregulation of estrogen response and proliferation genes, and upregulation of genes involving apoptosis, interferon response, KRAS, or tumor necrosis factor. The anti-tumor activity of the triple combination was associated with a decreased interaction between PR and ER. And finally, as a perspective, additional PDX models should be treated to identify predictive biomarkers of onapristone response, in particular to determine if PR expression itself is important for the activity of onapristone. Next slide. So I would like to thank the people in my group, Elodie, Ahmed, Léa, and Rania, in the hospital, the clinician, Guillaume and Paul Cotu, Muriel Romancé for the dual link essay at the Centre Léon Bérard, Pierre Delagrange and Ariane Joly, who performed the bioinformatic analysis of RNA-seq, and the context therapeutic. With them we collaborate on this project, and the patients that accepted to share their samples. Thank you.
Thank you very much, Elisabetta. And we do hope you find those predictive biomarkers. May I have the next slide, please? Our third presenter today is Dr. Tony Dussault of the Mayo Clinic. And Tony takes us into the arena of cancer cell stemness. And this refers to the stem cell-like phenotype of tumor cells that is poorly targeted by most standard therapies. The stemness properties of cancer stem cells sustain cancer progression over extended periods of time through enhanced capacities for quiescence, self-renewal cloning, drug resistance, regrowth, and the early emergence of metastasis. Dr. Tsoro will discuss the relationship between progesterone receptor activity and the key oncogenic signal axis associated with stemness. Tony?
Yes, thank you very much, Evan, for the kind introduction. So next slide, please. So I will show here our working hypothesis focusing on estrogen receptor positive breast tumors. In endocrine sensitive breast tumors, initially the majority of bulk cells exhibit a luminal CD44 low, CD24 high, LDH low phenotype that is sensitive to standard of care endocrine therapies. So during tumor progression, the aberrant activation of the Aurora A mitotic kinase may drive the enrichment of a population of cells showing a CD44 high, CD24 low, LDH high cancer stem cell-like phenotype that will be responsible for the resistance to endocrine therapies. This is because by lowering the ER expression, actually this cancer stem cell, or also more properly termed cancer initiating cell, may show a pan-endocrine resistance. So through the pharmacologic targeting of Aurora A, we can reverse this phenotype, so rewiring the cell to a more differentiated ER-positive phenotype that will regain sensitivity to endocrine therapies. Next slide, please. So to test our working hypothesis, first we use clinically relevant breast cancer models that were developed in the laboratory of Dr. Matt Getz at the Mayo Clinic. These are ER-positive MCF7, AC1 xenografts that show resistance to letrozole. However, when these cells were injected in tumor xenograft and treated with fulvestrant, they were actually highly sensitive to fulvestrant. So this mirrors the clinical scenario with patients. They are resistant to first-line endocrine therapy, but then they are sensitive to second line. However, the prolonged exposure of the xenograft to fulvestrant over time led to the selection actually of a clone of a subclone populations that were also resistant to fulvestrant. So first of all, to evaluate their cancer cell plasticity, we isolated cells from the letrozole-resistant xenograph, and this we term this ex vivo model MCF7LR, and from the letrozole and fulvestrant-resistant model, so we term this ex vivo model MCF7LR-FR. As you can see in this graph, when we perform FACS analysis using 2D adherent cell versus tertiary 3D models to generate mammospheres, there was no significant change in the enrichment of the CD44 positive, CD24 negative cancer stem-like population. However, the situation was different in the LRFR cells because from 2D versus the 3D mammosphere cultures, there was actually a shift where the majority of cells show a CD44 positive, CD24 negative phenotype. Next slide, please. So since we focus in our lab on the role of Aurora A kinase in the development of endocrine resistance, and also we have previously published the pivotal role of Aurora in the development of epithelial mesenchymal transition and also tumor stemness, we want to evaluate the difference in the expression of total and phosphorylated Aurora in these two models. So significantly, the LRFR models, both actually the 2D and the 3D mammospheres, show a higher phosphorylated Aurora A that represent the active form of the Aurora kinase. And there was this cell also show loss of ER. We also have previously published that high Aurora A activity is linked to loss of ER expression. And since cells that show high phosphorylated activity of Aurora A also are dependent on the Aurora A to grow, we developed tumor xenografts with the LRFR model. We corroborate the previous findings. This cell grow despite the treatment with the fulvestrant. However, when we combine fulvestrant with the Aurora inhibitor alisertib, we show the arrest of tumor growth. So these experiments also demonstrate that these cells, although they are endocrine therapy resistant, but they are highly sensitive to Aurora kinase inhibitor. Next slide, please. So now to define the mechanism, how Aurora, that is a mitotic kinase, promote endocrine therapy resistance and tumor stamens, we performed an unbiased phosphoproteomic approach where LRFR cells were treated with alisertib, or we use also the FDA-approved CDK4/6 inhibitor palbociclib also as control. And by performing bioinformatic analysis focusing on significant canonical oncogenic pathways, we show that these LRFR cells treated with alisertib, they have downregulation of IL-6 signaling. Then by performing a more granular analysis, we discovered that the key protein in the IL-6 signaling that was downphosphorylated by alisertib is the STAT3. So we showed that the treatment of the LRFR cell with alisertib reduced the phosphorylation of STAT3 at the serine 727 site. Next slide, please. So next step was to define now how the Aurora A STAT3 oncogenic axis promote self-renewal capacity. So first, we look at key stamens genes, KEL4, NOTCH2, and WNT, where LRFR cells were treated with a selective Aurora A inhibitors. So basically, we perform another Aurora A pharmacologic targeting and look at the gene expression by real-time quantitative RT-PCR. And we show there was a significant reduction of KEL4. So we engineer LRFR cell to express lentivectors with shRNA STAT3. Also, we show that genetic targeting of STAT3 reduce KEL4 expression. So this data demonstrate mechanistically that STAT3 expression is required to mediate Aurora A-induced KEL4 expression. And then we want to define the causative role of KEL4 in promoting the self-renewal capacity of endocrine therapy resistant cells. So we knock down also by using lentivector expresses shRNA targeting KEL4, and we show that genetic targeting of KEL4 reduce the self-renewal capacity of endocrine therapy resistant mammospheres. So overall, these experiments the study defined a novel oncogenic axis, Aurora A STAT3 KEL4, in promoting self-renewal capacity and endocrine therapy resistance. Next slide, please. So the significant now part of this study is that since we have completed at the Mayo Clinic a phase 2 clinical trial in metastatic ER-positive breast cancer patients treated with alisertib, this trial has been conducted by Dr. Tafay Haddad at the Mayo Clinic. We also have RNA-seq data and the unique PDX model. So we are now testing whether the patients that have been treated with alisertib, this Aurora inhibitor, also show an increased expression of IL-6 signaling. So in these experiments, we use the LRFR as a control, and we compare two patients that have been actually they were exposed to alisertib. One model developed acquired resistant to alisertib, and the 1695 model basically developed resistant after end of cycle 1. So what is significant here is that we show that there is actually an increase of expression of IL-6, CD44, STAT3, KEL4. These are all our genes and oncoproteins of interest in this pathway. Next slides, please. So the next study has been based on our collaboration with Dr. Carol Lang at the University of Minnesota, because what Dr. Lang demonstrated is that STAT3 and actually progesterone receptor interacts at the promoter level of KEL4 to control KEL4 gene expression. What also we have demonstrated is that Aurora A kinase is required for the co-recruitment of STAT3 MPR in the promoter of the KEL4 genes. So by starting our collaboration with the Context Therapeutics, now we want to assess whether the combination of Aurora target therapy with the antiprogestin, such as onapristone, may actually enhance the therapeutic response. This is very important because based on our completed phase 2 clinical trial, patients treated with alisertib, they actually have a better response after resistance to standard of care endocrine therapy and CDK4/6 inhibitors. However, resistance to alisertib still occurs. So now we are evaluating novel therapeutic strategy to overcome the resistance to Aurora target therapies. So by performing tertiary mammospheres, here we demonstrate that the LRFR model that show resistant to fulvestrant show a reduction, a significant reduction of mammosphere growth when we combine the Aurora inhibitor with onapristone. Next slide, please. So now we also develop in a different models, the BT474, where we overexpress Aurora A. So this is a unique model we develop in the lab. And first of all, we test in 3D mammosphere model if the force expression Aurora A promote increased phosphorylation STAT3 and also promote the phosphorylation of progesterone receptor at the serine 294 that basically is also targeted by onapristone. So in this model, as you can see here, the mammosphere for control, they have basically less phosphorylated STAT3 MPR compared to the BT474 Aurora A model. Then we perform also a real-time 3D mammosphere assay using the InQSight instrument. And remarkable, we demonstrated that in this model, they show resistance to both alisertib and onapristone as monotherapy. There is actually a significant reduction of mammosphere growth when alisertib is combined with onapristone. Next slide, please. So based on this work, we propose that the pharmacologic co-targeting of Aurora A and PR may enhance the suppression of KL4 expression and reducing stemness plasticity, and also in this case, reverse the endocrine therapy resistance. So next slides. So these are our ongoing studies in the lab. So we perform most of the mechanistic study in vitro. So now we will assess in vivo in preclinical ER-positive breast cancer model the combination of Aurora A target therapies with onapristone. We will use our endocrine-resistant breast cancer cell, MCF7 LRFR. We also develop in the lab a unique LRFR palbo-resistant model, and also the BT474 Aurora A models. And then we will use also unique PDX-derived tumor xenograft from the completed alisertib phase 2 clinical trial. Next slide, please. So I want to thank my colleagues at the Mayo Clinic, Dr. Ingold, Dr. Getz, Dr. Haddad, that she's conducted the phase 2 clinical trial, our biostatistician, Dr. Vera Schumann, also our colleague, Dr. Carol Lang, the collaboration with her has been key for us to the Aurora STAT3 PR interaction, and also the proteomics core facility at the Mayo Clinic and our funding source. Thank you very much.
Thanks very much for that great presentation. Our final presenter is Dr. Joseph Rucker. Joe is a co-founder of Integral Molecular in 2001, and Dr. Rucker's team is constantly challenged to create high-quality druggable antibodies. This is even more of a challenge when a targeted antigen has an almost twin-like family member or paralog. So Dr. Rucker will discuss the reasons for targeting Claudin-6, as well as design challenges and outcomes of that work. Joe?
Great, thank you, Evan, and it's great to be able to present our work today. Next slide, please. Let me give you first an overview of what I'm going to talk about today. I want to talk about Claudin-6 as a really interesting target for multiple cancer types. And it's a great cancer target, but has some unique challenges. And the next thing I will talk about is there are development of antibodies against Claudin-6, which are able to respond to these challenges. And then our essentially transformation of these Claudin-6 antibodies into Claudin-6 CD3 bispecifics, which are very, very potent killers of tumor cells. And from there, I will talk about where we are, what the current status of the project is, and where we're going with it. Next slide, please. So oncofetal proteins are proteins that are expressed, that are developmental proteins that are expressed on cancer tissues, but are not expressed on healthy adult tissue. Claudin-6 is a fantastic example of that. It's overexpressed in multiple cancer types with, I think, a particular interest to us in context with its overexpression in ovarian and endometrial cancers, but really showing no expression in any healthy tissue, a healthy ovarian or endometrial tissue. And this is true in other cancers as well. Next slide, please. However, there is a challenge with this. Although Claudin-6 is an oncofetal protein, it has a close relative, a paralog that is called Claudin-9. Claudin-9 is very similar to Claudin-6, and in its extracellular domains, it only differs by 3 amino acids. I should note that Claudin-6 and the whole Claudin family are proteins that are multi-spanner proteins with 4 transmembrane domains, so they have a complex structure and are involved in tight junctions. So the challenges with Claudin-6 are you have this close paralog, Claudin-9, and in fact there are other paralogs, Claudin-3 and Claudin-4, that are also similar, which you also have to be concerned with. You have these complex structures, and you also have proteins that are actually very highly conserved in mice. So there are many challenges to developing high quality antibodies. But given that its role in cancer and that it's expressed in multiple solid tumor types, especially ovarian and endometrial, the fact that it's an oncofetal protein with such clean expression profiles means that it's excellently suited for use in T-cell engaging bispecific antibodies, which I think are becoming increasingly popular as modalities for a number of oncology therapies. Next slide, please. So integral molecular our focus has always been on creating antibodies against these challenging types of proteins. And so we underwent an antibody discovery campaign against Claudin-6 with the focus on getting absolute specificity for Claudin-6, but no other Claudin paralogs. We were able to successfully do this, and our final molecules, 2 of which are shown here, show exquisite specificity for Claudin-6. These are real-time binding studies, so these are showing both association and then subsequent dissociation curves. You can see our two of our molecules, IM301, IM302, show very strong binding to Claudin-6, and you can see that these are not only strong binders, but they're showing very, very slow dissociation. So these are very, very tight binding proteins. You see no binding to Claudin-9 and minimal to no binding to Claudin-4. However, there are other antibodies against Claudin-6 that have been worked on. There is a benchmark antibody, one of which, this one here, is one that has actually seen the clinic. Although it binds Claudin-9 well and relatively tightly, it shows substantial binding to Claudin-9 and even detectable binding to Claudin-4. And this off-target binding to Claudins such as 9 and 4, which are ubiquitously expressed, or expressed, not ubiquitously, expressed in multiple tissue types, in adult tissue, healthy tissue types, means that there are many potential liabilities as you go through the development process. So the antibodies we have developed have this absolute specificity. Next slide, please. So with that, we, starting with, working with Context Therapeutics, we began to build out our Claudin-6 antibodies into a CD3 bispecifics. And so what we did is we took multiple Claudin, integral molecular Claudin-6 antibodies. We paired them with a variety of well-characterized anti-CD3 antibodies. And the idea here is that you can bring T cells close to the tumor so you can get effective tumor killing. And so what we did is we took, multiple Claudin-6 arms, multiple CD3 arms, and we built them into a variety of different formats. And these formats differ by stoichiometry, affinity, specificity, geometry, size, functionality. And the idea is that we want diversity. We want the biology to tell us what to do rather than making any a priori preconceptions about what will work best. So we, made these library. We had we whittled it down to 54 bispecific candidates, which we put through a full array of binding assays, as well as T-cell killing assays and and then whittled it down to a smaller candidate panel. Next slide. So one thing I will note is we have a number of different bispecific formats. None of these formats change the specificity that we see with the parental Claudin-6 antibodies. This is an example here where we have once again, this is on Claudin-9 reactivity. The clinical benchmark shows strong binding, but all of the antibodies in in the different bispecific formats are really showing no binding to Claudin-9. Next slide. As mentioned, we took all of those 54 original antibodies, ran them through T-cell killing assays against both cancer cell lines as well as overexpressing cells, and whittled it down to a panel of 12 candidate antibodies. These antibodies show robust T-cell dependent cytotoxicity. We have multiple formats that are presented here, each with different Claudin-6 and CD3 arms. And this diverse panel is what we are then going to move. We're going to move forward with into in vivo studies. Next slide, please. Let me tell you the current, summarize and give you the current status of this project. We have completed this in vitro characterization of 54 bispecifics. They're diverse across the backbones and the sequences and the structures. They retain the selectivity of Claudin-6 over 9 and 3 and 4 as well. And they are potent killers, with some of them having cytotoxicity EC50s in the picomolar range. We've whittled this down to a candidate panel of 12 bispecifics, and this is currently undergoing scale up. We'll undergo additional developability testing, which will then lead directly into in vivo studies that will enable us to choose a lead candidate and backup candidates. So I want to thank all my coworkers at Integral Molecular who have helped move this project along, as well as NIH and especially Context Therapeutics for the resources to help move these projects forward. And thank you very much for your attention.
Thank you, Joe. Thank you. We can go to the next slide. I guess that's me. So we can go one more slide. We've heard a lot of great talks. Let me quickly summarize them. Soon to be Dr. Lauren Warner is discussed how onapristone facilitates breast cancer cell transition from immunologically cold to hot. And we certainly hope that this is going to lead to a combination of progesterone receptor inhibition with checkpoint inhibitors in the clinic. Dr. Marangoni discussed how onapristone enhances the therapeutic activity of anti-estrogens, CDK4/6s, and P1-3K inhibitors in PDX models of breast cancer. Dr. Tony DeSoro discussed pharmacologic targeting of the aurora kinase STAT3 progesterone receptor oncogenic axis, and how it impairs self-renewal capacity of breast cancer cells. So giving us a potential approach to resistance to anti-estrogens or CDK4/6 inhibitor therapy. Finally, Dr. Joe Rucker changed it up by switching over to looking at our bispecific program and giving us the first public presentation of Claudin-6 by CD3 bispecific data, highlighting remarkable Claudin-6 selectivity as well as effective T-cell activation. With that, we have a few questions in, and we're going to let me pass those on to some of our speakers. Let me just see if I can post this question, if everyone now sees this. So to Lauren. Emily Bodmer asked, as you mentioned in the presentation, breast cancer patients tend to be immune cold. How do you think this would translate when treating humans, considering onapristone activity was not as good when mice were immunodeficient?
That's a really good question. So you're totally right that breast cancers are immune cold. And the overall goal in doing that experiment with the immunodeficient mice was just to show that you need an intact immune system for onapristone to have its full effect. So I think if anything, that just kind of further serves to prove that our hypothesis may be correct, that you could use onapristone to convert immune cold tumors to immune hot tumors, because the activity of it depends on the immune system.
Thank you. And perhaps you mentioned that several other laboratories have contributed to this area, also finding that immunocompetence is an important measure of whether we can see this type of activity from progesterone receptor inhibitors, onapristone and mifepristone. Second question is for Elisabetta Marangoni. Let me see if I can post this. What do you think is the reasoning for why onapristone is effective when combined with estrogen receptor blocking agents, but not alone even in progesterone receptor high models? Elisabetta, you're muted.
Oh, sorry. Can you hear me now? So I was saying, I think it's an interesting question. I think one reason could be that these tumors are not only strongly dependent on PR, but also on ER. And we see that they are responding to ER targeting therapy such as fulvestrant. So I think that the double combination, the triple combination work because you target in the same time ER signaling and PR signaling. But when you target only PR signaling, you still have ER-dependent proliferation independently of PR. So what we clearly see is that onapristone alone is not sufficient to really have a strong effect. But again, this was only observed in 2 tumors. So I'm not saying that it's the case, always the case. We might have some tumors that are less dependent on ER and more dependent on PR, but at least in the first model that we treated, it seems that the combination it's really more effective than the monotherapy.
Thank you. Marty, would you like to reflect back on the activity of onapristone in the late 1990s, 2000 studies when it was used clinically?
Absolutely, and thanks for Evan moderating a great discussion. You know, I think as many of you on the Zoom are aware, onapristone was first evaluated clinically in the 1990s. And I think what caught all of our attention and why we're so excited about the drug is in patients with de novo or locally metastatically advanced breast cancer, the majority of patients in that study responded quite well to onapristone. So about 56% of those patients had their tumor shrink, what we refer to as partial responses. And the durability of that response was quite prolonged, almost 18 months. So clearly something interesting is going on when the drug is used as a monotherapy in patients who do have intact immune systems. And I think we heard a lot today, and a really critical piece of the discussion was the importance of an immune system and understanding that these hormone-dependent tumors historically are cold and have had very little responsivity to checkpoint inhibitors or other immune-targeted agents. But clearly something goes on here where we combine onapristone with an intact immune system like the historical studies, or in these more recent preclinical studies. And you see some really interesting things happen, at least in mice. So we're very much looking forward to, as Dr. Marangoni pointed out, the clinical evaluation of this oral progesterone receptor antagonist in combination with anti-estrogen, something that we've seen really nice data preclinically, and we're evaluating across a variety of female cancers in the metastatic setting.
Very good. Well, on behalf of myself and Marty and our colleagues at Context, let me thank the presenters. Let me thank the folks who have come to listen to today's presenters. And I'll pass this back to Sue.
That's all we have time for today. Thank you everyone for submitting your questions, and thank you for taking the time out of your busy schedule to join us on this call. If you have any questions, please feel free to reach out to us directly. Our contact information is listed on the Context Therapeutics press release, and we'd be happy to talk to anyone with additional queries. Thank you, everyone. Bye.